So module 6 is going to have 3 chapters, 16, 19, and gram-positive and gram-negative pathogens. And the it will be another one of those tests with 80 questions and 160 minutes. So it's another one of the long tests. What I have here is chapter 16, which we're going to start covering it, and we may be able to cover it all, we'll see. If not, we'll finish it next time, because 19 is actually short and easy. So we're having a longer chapter 16, short chapter 19, and then the other 2. Okay, now 16, the way I look at it, 16 is the flip side of the coin of chapters 14 and 15. 14 and 15, we looked at how our immune system defended itself from microorganisms, specifically bacteria, and viruses too, obviously. But chapter 16 is going to look at how bacteria, especially, can establish an infection overriding the immune system. What is the counterattack, if you will, of the bacteria, or how bacteria can protect itself from our immune system. So first thing is that when we talk about infection, we're really referring to, so when we talk about a bacterial infection, we're really referring to a pathogenic type of relationship between a host and the bacteria. Because when it comes to infection, we are already infected with bacteria. We're infected with millions, billions, and billions of bacteria that already live in our bodies. And we live in a symbiotic relationship with them, in which both of us benefit from that relationship. This is what we call the resident microbiota, the resident flora. They are the permanent guests, versus the visitors, which oftentimes are the ones that become pathogenic. Those are the transient microbiota, temporary guests. These are the ones we remove when we take a shower or when we wash our hands. Okay, so pathogenic bacterial infections can come on 2 types. Most of the time is the result of a normal flora that has either overgrown or it's appearing in the wrong place at the wrong time. So those are the 2 scenarios when normal flora causes infections, and most infections are normal flora infections. Okay. Sometimes, however, true pathogens will cause an infection. So the first thing we need to do is to have an idea of what kinds of organisms are normal on every part of the body that does have a normal flora. So that as a medical microbiologist, you can take a look at the organisms growing from a specimen, and you can say, no, this is normal, or you can say, no, this is not normal, or this organism doesn't belong here. Okay, so you need to know, especially the organisms that are in red mentioned in this chapter. So the skin is an easy one, because most of the skin is covered by one organism, Staphylococcus epidermidis. The skin, the epidermis of the skin, is not a propitious place for microorganisms to grow. On the contrary, go back to chapter 14. What makes the skin a good barrier that doesn't allow the growth of microorganisms, or at least the going through the skin by microorganisms? So not very many organisms can withstand living in the skin. Staphylococcus epidermidis has very successfully colonized the entire skin. In specific places like sebaceous glands, we find organisms like Propionibacterium agnes. And then Staphylococcus aureus is in some places on the skin, typically more humid places, under the arms, in the genital area, in the nose. In the nose, we're going to find the same organisms we find in the skin, plus more of the Staph aureus, for example. So Staph aureus is actually a place where the nose, the nasal cavity, is one place where oftentimes Staphylococcus aureus colonizes. Now, the throat is another place where we find a lot of microbial growth. And in contrast with the skin, we find a lot of variety in the throat. It is a really good place for microbial growth. It is moist, warm, lots of places to hide, variety of oxygen requirements, oxygen environments. So there's a lot of organisms that grow in the throat. Many of them, there's tons of different, well, I wouldn't say tons, lots of different species of Streptococcus. This is a really good place for Streptococcus. This is a picky organism, aerotolerant, doesn't do extremely well in any environment, but especially it's not crazy about oxygen, it's good that way. Corynebacterium is a very successful organism pretty much all over the body, so that's no surprise. Neisseria, yes, absolutely, as in gonorrhea, yes. But in this case, this would be other Neisseria species. Windows here. Okay, so yes, Neisseria is in gonorrhea, but in this case, typically this species is actually meningitis. Neisseria meningitis is common in pretty much all of our throats. It's normal flora, even though it does cause meningitis, as long as it doesn't overgrow and it's not a pathogenic, particularly bad pathogenic strain, we're okay. Lots of Neisseria in the throat also, though. Let's see, so Haemophilus influenza, that's another one. Now, a lot of these organisms are going to begin to colonize our throats as we grow older. So a young baby will not have been colonized by these organisms. And that's why they're more susceptible to things like Haemophilus influenza causing meningitis, or Neisseria for that matter, or Streptococcus pneumonia. But by the time we're adults, our throats are teeming with all kinds of microorganisms. You can see here a normal flora plate of the throat. And you can see how there's many different, at least you can see 3, 4, 5 different types of organisms growing in there. Okay, so another ones in red, those are the important ones. And we'll talk more about some of them, like Streptococcus pneumonia and Streptococcus pyogenes, both of whom could be pathogenic, could cause infections. All right, so that's the throat. Another good place for microorganisms is the mouth. Lots of Streptococcus, again, lots of places to hide, lots of food, lots of moisture. The only bad thing about the mouth is that it's constantly being washed by saliva. Other than that, it's really a good place for microorganisms. Lots of anaerobes, which you wouldn't think about it in the mouth, but there's enough places to hide to where strict anaerobes can live there very, very happily. So Fusobacterium, Leptotrichia, Bacteroides, all of these organisms are common in the mouth, and the ones with asterisks are the anaerobes. We are learning more and more about normal flora, and there's more and more of a link between the normal flora organisms in our bodies, even something as odd as the mouth or the GI tract, and diseases like Alzheimer's, diabetes, type 2 diabetes, overeating. So the more we know about the normal flora and the kinds of organisms that may begin to overgrow, and the link to those organisms to certain conditions, the more important it becomes to be aware of the kind of normal flora that's growing in our bodies. And normal flora of the mouth does have a link, especially to Alzheimer's, actually. Not in all kinds of Alzheimer's, but in some types of Alzheimer's. Okay. Large intestine, yeah, this is probably the place where we have the most microorganisms. Tons and tons of microorganisms. The most abundant type are the strict anaerobes. A lot of the same anaerobes that we find in the mouth, we're going to also find in the large intestine. Bacteroides, Clostridium. The second most abundant will be the members of the Enterobacteriaceae family, like E. coli, Proteus, Enterococcus, etc. other organisms, lactobacillus, enterococcus, streptococcus, pseudomonas, some yeast. So the normal flora in the large intestine is extremely varied. Urethra, actually, it may even vary between males and females, but in general, lots of streptococcus, mycobacterium, especially in males, bacteroides, which is an anaerobe also found in the mouth and the large intestine. Vagina, that's a very special place. It is loaded with a bacteria called lactobacillus. Lactobacillus is the same bacteria responsible for some of the acidic foods that we eat, like pickles or even yogurt. Yogurt, absolutely, even yogurt, yep. The bacteria ferments, and when it does that, it produces lots of acids. So that's what it's doing in the vagina. It's creating an acidic environment that would prevent the growth of other microorganisms. There's also some yeast, some streptococcus, but lactobacillus keeps the normal flora in check so that nothing overgrows due to the acidity that it produces. So the normal flora is established during birth, and the composition of the normal flora changes over time. So when a baby is born, as it's going through the birth canal, it's acquiring its first normal flora. The baby inside the womb is sterile. That's a completely sterile environment. So the first encounter that the baby has with bacteria is as it's going through the birth canal. What we know now is that babies that are born through cesarean do not end up with the same normal flora colonization as babies born through a birth canal. And those babies, the babies born by cesarean, later on may be more prone to things like asthma or allergic reactions. So knowing that, nowadays, sometimes the doctors are taking samples from the baby, from the mother's birth canal, and smearing the baby with that bacteria as if the baby is being born by cesarean section, just because we're beginning to understand the importance of establishing a normal flora. Something else that we're doing that, which it's becoming a very common practice, is to have stool transplants, fecal transplants, so that if somebody has a normal flora in the large intestine, yeah, I know it sounds horrible, but it's being done, and it's being done quite commonly it's nothing that is necessarily fancy in large medical centers. It's being done in regular clinics. If a patient has a recurrent infection, intestinal infection, due to an overgrown of a normal flora, they're beginning to do that. They're beginning to do fecal transplants, and it works very well. I know it's yucky, but whatever works, and I'm sure it's not very expensive either. Okay, wonderful. That should end you. Oh, interesting. You feel okay? Yeah, I'm telling you, it's not strange at all. And in the United States, we are not using it for very. It's only being used to repair, if you will, normal flora in the intestines that it's off. In Europe, it's being used for other reasons. Like again, try to lose weight if somebody's chronically obese, and sometimes the normal flora of the intestine has a lot to do with it. And so it's just a matter of eliminating the bad normal flora and giving the person healthier normal flora. So yeah. Anyway, so it's a big field of study, normal flora on our bodies. So what does it do? It's definitely protective. It protects against harmful organisms by what's called competitive exclusion. Essentially, any other organism that tries to colonize the area will have to compete with our normal flora for food and moisture and space. On top of that, it is constantly stimulating our immune system, which is again very, very good. So those are the two means through which normal flora is helping us. So this is definitely a symbiotic relationship where both being benefited from the relationship. Okay, so, oh, yeah, few things. Know the places in the body that have normal flora. We've mentioned those. Know the most abundant normal flora in these places. Um, know how normal flora protect us. Also, be aware of the places in the body that do not have normal flora, which will be the ones we did not mention. We did not mention the central nervous system, the heart, blood, lungs. Okay, those are the ones that come to mind off the top of my head. Kidneys, liver. So any place we did not mention is a place that should not have normal flora. Okay, so we talked about we are already infected with microorganisms, but the kind of infections that harm us is the one that is a parasitic relationship with the microbe. Oftentimes, microbes establish a subclinical infection. This is one in which there are no overt signs or symptoms of an infection because they're very, very mild. So they go unnoticed for a long time. STDs are oftentimes subclinical infections in females. Gonorrhea and chlamydia are oftentimes asymptomatic in females. So that's what a subclinical infection is. It's an asymptomatic infection. So be able to know or to give examples of infections that are subclinical or can be subclinical. All right, make sure you can tell the difference. So is it just not? It is not noticed by the patient. Oftentimes not at all until there is a obvious damage. So oftentimes the damage is so mild or so hidden that the patient does not notice. So think about that. In a female, if you have discharge, you're not going to think much of anything. Maybe you're close to the menstrual cycle, and there is normal discharge throughout a cycle. So that may not be anything alarming to just have some discharge. So it will go unnoticed. Whereas in males, a discharge will be noticed, and it will be cause for alarm. It is not until the damage is painful or begins to cause other symptoms that it will be noticeable. So it's a mixture of both. If it does tests, yes, but a provider may only be able to go by what the patient is feeling. But if it runs tests on the discharge, if there is some inflammation, but even that may go unnoticed. So this is so common that in females, STDs are oftentimes symptomatic that routinely every baby that is born, it is given in the eyes little drops of antibiotics to prevent ophthalmic gonorrhea. That is how prevalent it is that they don't even bother testing the mother. They just automatically across the board, which again, this is just a public health strategy. That way we catch everybody, and a few drops of antibiotics in the eyes of a newborn are probably not going to hurt much of anything, whether they have something or not. And if there is something, then definitely will help. So that's how prevalent it is. I would say how aware the medical community or the public health community is of the problem. Okay. So yeah. Definitely know the difference between signs and symptoms. Symptoms are experienced, meaning they are subjective. You cannot measure them. So this will be something, or at least you cannot measure them objectively. So there can be something like pain. It could be, can't think of anything about pain right now. It could be dizziness, tingling. Oh, good. Yeah, it could be a tingling. It could be a dizziness. It could be pain. So again, something that is typically subjective. A sign is something that can definitely be observed. Is objective. I mean, symptoms are subjective. Signs are objective. They can be measured, they can be examined. So that would be fever, again, a discharge, a redness, etc. High blood pressure. So know the difference between signs and symptoms and be able to identify what is a sign, what is a symptom. Primary infections versus secondary. Primary infections are the initial disease, which are often viral in nature. The initial infection, the primary infection, weakens the immune system, opening the door for a secondary infection. Secondary infection is typically bacterial in nature, and that is the one that is really bad and oftentimes kills the patient. So that is the scenario, that's the cases with things like influenza and pneumonia, viral pneumonia, I should say, even viral meningitis for that matter. So these viral infections weaken the patient's immune system, and that opens the door to bacterial pneumonia or bacterial meningitis or a bacterial infection, and that's oftentimes what is a lot more serious. Pathogenicity is the ability of organisms to cause disease in healthy people. So a true pathogen will be one that causes disease across the board, not just in immunocompromised or weak hosts, but in anybody. So rabies, measles, tetanus are examples of true pathogens. Versus opportunistic pathogens. And this is the deal. This is one of the keys between a pathogen and an opportunistic pathogen. An opportunistic pathogen is common in the environment or as a normal flora. A true pathogen is not common. The rabies virus is not common. Measles, nowadays especially, is not common. Tetanus is not common. On the other hand, E. coli, it's incredibly common. We're probably exposed to E. coli every single day. Staphylococcus aureus or Staphylococcus epidermidis, both of them can be opportunistic pathogens. They're incredibly common. Pseudomonas aeruginosa, incredibly common. Okay, they will cause disease typically in a weak host or when they manage to go in a place where they shouldn't be. A degree of pathogenicity is called virulence. So the more virulent an organism, the more pathogenic it is, the more able it is to cause disease. So highly virulent organisms means a high degree of pathogenicity. When we go into gram-positive pathogens, we will talk about Streptococcus pyogenes. Go back to normal flora. This is one of the normal flora of the throat. This is a highly virulent organism. We will actually, even in this chapter, we'll start talking about the features that the organism has that allows it to overcome our immune system. So it's extremely pathogenic, very virulent because of that. However, it doesn't compete very well with normal flora, even though sometimes it is normal flora, but even at that, it doesn't compete very well. Very picky, very hard to grow. So this is an organism that is very virulent, and however, it can be fought off fairly easy if we have a good, healthy immune system. So the key to virulence is not only, or the key to establishing an infection is not only the degree of virulence, but also the numbers. If you are infected with a few organisms and those few organisms are extremely virulent, then there's definitely something to worry about. If you're infected with a few organisms, but they're not very virulent, then chances are there's nothing to worry about. Okay, so it is a play of not only virulence, but numbers. Yeah, I did not talk about infectious dose. It comes, okay, good. So I don't know why that question is right there, but okay. All right, then we'll address it when it gets here. Okay. Communicable diseases, contagious diseases. All right. The book doesn't make much of a difference anymore, or the differences are not as pronounced anymore. So I used to spend more time on these than I do now, because I realized the book is not really distinguishing them very well anymore, which is fine. This is the deal. A communicable disease is one that that can be spread. Okay, that is pretty self-explanatory. A contagious disease is a communicable disease that is easily spread. That's the difference. So the hepatitis, well, this is the acronym is backwards. It should be HBV, hepatitis B virus. It is communicable, but it's not very contagious. So it can spread, but it doesn't spread easily. We get it through blood. We get it through sexual contact. We get it through sharing of needles. Those activities are not very easy to engage in. Contrast that with chickenpox. Chickenpox is contagious. It is easily communicable. You just have to be close to somebody who has chickenpox, and you may get it. Okay, so all it requires proximity. Okay, so that's the difference. HIV is communicable, but it's not contagious. It's not easily communicable. So that's basically the difference between communicable and contagious. So contagious is communicable, but it's easily communicable. Infectious dose is the minimum number of organisms that can cause an infection. The lower the infectious dose, the more easily the organism is spread. So that means it's more pathogenic, it's more virulent. Think about it. If 3 organisms can cause an infection, that will be more virulent than if you need 100 to cause an infection. So an organism that has a low infectious dose, there you go. Low dose, high pathogenicity. Exactly. There you go. So it's an inverse relationship. An organism that has a low infectious dose is a lot more pathogenic than one with a high infectious dose. Rhinovirus has a low infectious dose. 1 virus can cause an infection. Rhinovirus is the virus that causes the common cold. Salmonella has a very high infectious dose in the hundreds. So you cannot get salmonellosis very easily. It's not as pathogenic. Okay, stages of infectious diseases. Incubation is the time between the exposure to the pathogen at the onset of the symptoms. And there are many factors that influence incubation period. So there's never a set incubation period for all diseases. Each one has its own infectious incubation period, and even that depends on many other factors. Once the illness, there's the onset of the illness that follows the incubation period. That's when the individual experiences the signs and symptoms that are typical of that disease. Right between these 2 stages, infection, the incubation and the illness, sometimes the patient experiences prodromal symptoms. And prodromal symptoms are the very, very, very early symptoms. Notice that they're called symptoms, not signs, because they're typically something that the patient feels, but it cannot be measured. So you feel weak, you feel like you're coming down with something. Those are prodromal symptoms. After the patient has gotten over the illness, then he goes into the period of convalescence, of the recovery from the illness. A patient is typically considered infectious in all 3 stages, even if technically it may not be, it's just considered infectious in all 3 stages. So when you look at these three stages of an illness, of an infectious disease, you can distinguish between acute, chronic, and latent types of infections. An acute infection has all three stages pretty well delineated, one after the other. Versus a chronic one, which may have an incubation period, and then the illness is very, very, very long, and oftentimes there is no convalescence because the illness either never goes away, or oftentimes is the cause of death of the patient. Latent repeats these cycles over and over again. So incubation, I guess, except for incubation, illness, convalescence. Then there is a period of no illness, and then we begin to, again, illness, convalescence. Then no illness, and then again, illness, convalescence, and that continues to repeat. Localized versus systemic infection. Localized is the infection in a small area versus systemic, which is generalized, is spread through the body. An infection typically will spread through blood or lymph. Toxemia is when we have the toxin products of a microorganism circulating in the blood. Viremia is when viruses are circulating in the blood. Septicemia is very serious, is when we have both the infectious agent itself and the products circulating in the blood. Okay. And then this last part looks at what's called Koch's postulates, which are a series of steps that were established to determine that a specific microorganism was responsible for a particular disease. And these postulates were put together by Koch, who was a German physician of the turn of the of the 19th century. So late 1800s, beginning 1900s. He was a contemporary of Pasteur, and he and Pasteur were trying to show that bacteria could cause disease, and he's the one who ended up showing it conclusively. So in order to show that bacteria can cause disease, he created this series of steps that are geared to proving that, or to showing that organism A causes disease A. Now, these are not perfect, but they are useful. And as long as we're aware of the imperfections, then Koch's postulates are very useful, and they're still being used to this day. So he was working with anthrax, with a bacteria called Bacillus anthracis that was causing a disease called anthrax, which is mainly a disease of animals, but it could also be transmitted to humans. So this is what he did. He took subjects, animals that had died of the disease, and every time he isolated the same bacteria. Then, once he isolated the bacteria, he noticed that the bacteria appeared to be the same on every case of the disease. He took a laboratory host and injected the bacteria into the host. The host then came down with the disease and died. And when they took bacteria from the dead subject, the bacteria isolated was the same as that had been isolated from the other animals that had died from the disease. Okay, so he did this many, many times, and he enumerated them, the steps. So the suspected germ must be present in every case of the disease. So every single individual that's diagnosed with anthrax and it comes down with anthrax should have the Bacillus anthracis in their blood. Once the bacillus or the bacteria is isolated, it should be able to give it to a laboratory animal. Laboratory animal should come down with the same disease as the original subjects, and the same organism should be isolated from the laboratory animal. Okay, so those are Koch's postulates. Now let's talk about the problems of Koch's postulates. First of all, is the first one. It says microorganisms must be present in every case of the disease. He was thinking one microorganism, one disease. We know now that that is not the case. We know now that one microorganism could cause many different diseases. So, for example, Streptococcus pyogenes can cause strep throat. It can cause scarlet fever. That's the only two that can come to mind right now. But there's impetigo. It can cause skin infections. Streptococcus pneumonia, for that matter, can cause pneumonia, can cause meningitis. Okay, so there's not such. Oftentimes, we don't have one microorganism, one disease relationship. The opposite is also true. We can have one disease being caused by more than one microorganism. So we have disease and many microorganisms that could cause it. Case in point, meningitis. The three most common causes of meningitis: Neisseria meningitidis, Streptococcus pneumonia, and Haemophilus influenza. So one disease, many microorganisms that could cause it. So there isn't this one-to-one relationship. So when we go back here, and it says every case of the disease, we should end up with the same organism, that may not be the case. If the disease is meningitis, you could have 3 different microorganisms isolated from each one of those hosts. Okay, the microorganism must be grown in pure culture from the disease host. Again, that may not be possible because some microorganisms are cannot be grown outside the human host. For example, the organism that causes syphilis has not yet been grown in the laboratory. The organism that causes leprosy has not been grown in the laboratory. The organism that causes tuberculosis is very hard to grow in the laboratory. So we would not be able to do this right here. And then the last postulate, the same disease must be produced in susceptible experimental hosts. Well, sometimes there are ethical considerations. So organisms can only be grown in humans, and so you're not going to infect a human on purpose. So that will not be possible. That last step will not be possible to show. Another problem is that some hosts may have the organism, but they may not end up with the disease. So we have asymptomatic carriers of an organism, which carry the organism, but for whatever reason do not come down with the disease. Typhoid Mary, case in point, was an asymptomatic carrier of Salmonella typhi. She gave typhoid fever to hundreds of people. She never had the disease. Okay. Well, she carried it, but she never came down from the disease. She never had it, but she carried the organism. So be able to explain the drawbacks of Koch's postulates. You may hear at some point or another, HIV, it's never been proven that the HIV virus causes AIDS. And that is true because we have never done Koch's postulates with the HIV virus because of ethical considerations. But there are other means to show that a particular virus or microorganism causes a disease that doesn't necessarily involve Koch's postulates. So we're actually trying to go more for molecular postulates in which we show that the gene that causes the disease in the organism is the one that's present, not the entire organism, for example. So, but you still need to know Koch's postulates and what they say and what the drawbacks are. Okay, any questions? Correct. So virulence genes are the ones that make make the organism pathogenic. So a virulence genes would be one that codes for a toxin or a capsule, for example. So and even you can just take the gene itself and give it to another organism that is not virulent, and you can make it virulent. Absolutely, yes. Oftentimes, virulence genes are found in plasmids. many, many times, especially in the case of toxins. So an organism could acquire a virulent gene by just acquiring a plasmid. So we're trying to look at the genes that make an organism pathogenic as the way to correlate the organism with the disease. Okay, so the second part of this chapter looks at more specifically at what kinds of features microorganisms have that help them to cause disease. So first thing that a microorganism must do, and we're focusing on bacteria more than anything, the first thing the bacteria need to do is to attach to cells in our bodies. Adherence, that's the first thing. So it has to be able to breach barriers or even go through the barrier, use the barriers within the barriers themselves to attach. Okay. So microorganisms, bacteria, attach to their host cells by using adhesins, which are proteins that are found on the surface of the bacterial cells. So these are adhesins proteins. They can be found in the pili, for example, at the end of the pila or the fimbria, or even as part of capsules of a bacterial cell. So this is a bacterial cell that has fimbria. I guess they're calling it pili now, but okay, pili or fimbria would be a little bit shorter perhaps. And at the very end of this protein structure, there is a molecule called adhesin with which this bacterial cell binds to specific receptors in the host cell. So this is the cell membrane of the host cell. That will be the host cell there. That will be the nucleus of the host cell. And the bacteria is binding to receptors in the host cell. Okay. All right. This specific binding dictates what kind of cells the bacteria can attach to and what kind of disease it will cause. Oftentimes, that will determine whether the organism is pathogenic versus non-pathogenic. Pathogenic one may be able to bind, a non-pathogenic one may not be able to bind, for example. So the next question would be, how does the organism protect itself, or how does our immune system, protect itself from adhesins? Well, one of the ways is by making antibodies against the adhesins and covering the adhesins with antibodies, as in immobilization, for example, and prevention of adherence. We saw these in chapter 15. Okay, so this is what our immune system does. This is chapter 15. The next question is, how does the microorganism fight off immobilization and prevention of adherence? So what can the microorganism do? Okay, it can have a rapid turnover of pili, it can have antigenic variations, or it can make IgA proteases, or it can do all three actually. Now, let's start down here, which is the easiest one. These antibodies right here are IgAs because they are found in mucous membranes. Remember, this is what happens at the very beginning when the organism is trying to gain access to our body. Organisms gain access to our body by going through mucous membranes. Remember, the most common mucous membrane, the number one portal of entry is the respiratory tract. So this could be a respiratory tract bacteria that's trying to gain access to deeper places in the body. And in the respiratory tract, we'll have mucus and saliva that is loaded with these IgAs that will bind to the adhesins in the bacteria. And with that, it will immobilize it and prevent adherence. Okay, the bacteria in turn could secrete IgA proteases. IgA proteases will be enzymes that destroy proteins, proteases, more specifically IgAs proteins. So that will destroy the antibodies and it will prevent the antibodies from immobilizing the bacteria. So there is one strategy. The other strategy is rapid turnover of pili. Okay, let's go for that one now. Rapid turnover of pili essentially means, and actually kind of goes together with antigenic variations. What if the organism sheds the pili? Okay, so if it sheds the pili, now it's going to shed the pili with the antibody attached to it. And you're going to say, well, now it doesn't have the pili. True, now it doesn't have the pili. No big deal. Let's grow another set of pili. And what if, like a lizard's tail, there you go, like a lizard's tail, exactly, except that this is probably even more efficient because it's simpler. And what if the new set of pili has antigenic variations? It's just different enough from the previous set of pili that the antibodies now cannot bind to it. And now our immune system has to go through the trouble of creating new antibodies against that new pili, which, as you know now, that could take as long as 2 weeks. Okay, so there you go. Those are some good strategies that the bacteria can follow to prevent being stopped by antibodies from gaining access to the host. Antigenic variations, IgA proteases, rapid turnover of pili, Neisseria gonorrhea can do all of these. And actually, Neisseria meningitis can do the same thing. Neisseria is a genus of bacteria that are very, very successful at gaining access to our bodies through mucous membranes. And the reason why is because they make IgA proteases, they have antigenic variations, and rapid turnover of pili. So that's why Neisseria gonorrhea and Neisseria meningitis are very successful at that. Okay. So let's say the organism is successful at gaining access to our cells. Now it has to invade. If it stays in the mucous membranes, it's probably not going to be there for very long. Mucous membranes, again, has a lot of protection. They're typically being washed by saliva or urine or food. So it's not a place where the microorganism typically can stay for very long. So it has to go deeper in the body. It has to invade. All right, so how can it do that? Colonization. A few strategies. Use type 3 secretion systems. Gram-negatives do these. Exploring antigen sampling cells, and then take advantage of trauma or vectors to go through skin or even through mucous membranes for that matter. Type 3 secretion systems and exploding antigen sampling cells, those would be strategies in mucous membranes. Take advantage of trauma or vectors, that would be strategies to go through the skin. All right, so let's talk about going through the skin. Not very easy, most difficult barrier to penetrate. And bacteria that goes through this route has to destroy the skin. So it may use a vector like a mosquito or a flea, or it might be just an accident, like a wound, and the wound is utilized to go into deeper areas. Okay, most common route of entry, the respiratory tract. Now, next one is causing direct uptake by host by delivering type 3 secretion system. That is, or exploring antigen sampling processes. Let's go for exploding of antigen sampling. Okay, this is what it is. And this happens especially in the GI tract. In the GI tract, we have cells that their only job is to sample antigens on their surface and take these samples to deeper tissue underneath. These are called M cells, and M cells are constantly eating up, if you will, stuff that's found on the surface of the GI tract. And that stuff is presented to the immune system. The immune system will react, and remember, one of the things that bacteria do is to keep our immune system in check, always working. Some bacteria take advantage of that. So they are taken up by the M cell. They live, survive the M cell, and is able to cross to the other side. Remember, this is a mucous membrane. So this is a tract, like the GI tract. And now it goes to the other side. Yes, the M cells are different from dendritic cells. They're part of the epithelial lining. So this is part of the epithelial lining of the mucous membrane. And so the bacterial cell now goes to the other side, where we're going to find the second line of defense, which are the phagocytes. And the phagocyte will take it up and eat it. However, the bacteria will be able to survive the phagocytosis, escape the phagocyte, and now begins to invade other cells from inside. And oftentimes it stays inside cells, moving from one to another, because that's safer. And that way escapes the immune system if it is hidden inside cells. So that's a strategy. Shigella does this. Shigella is a bacteria that causes a disease called dysentery, infects the GI tract, and that's how it invades, is by taking advantage of the M cells, and then it stays pretty much in the lining, jumping from one cell to the next. And it causes diarrhea, actually causes bloody diarrhea. So that is strategy. Type 3 secretion systems, this is what they are. And we have seen type 3 secretion systems in gram-negatives that invade the GI tract. So again, we have the GI tract. That's not the only place, but that's one of the places. The bacterial cell comes along and lands on the surface of the cell. Once it does that, it literally creates this little protein injection that is going to go through the bacterial cell, and is going to inject a series of proteins inside the bacterial cell. Those proteins are going to rearrange the cytoskeleton of the cell and make the cell eat up the bacteria. So now the cell phagocytizes the bacteria. Remember, the goal here is to go into deeper tissue. Now the bacteria has been phagocytized. So it's inside the phagosome. And inside the phagosome, the bacteria will again secrete type 3 secretion systems, proteins, which now these proteins are going to surround the phagosome and prevent phagosome-lysosome fusion. And with that, there is no danger of being killed inside the bacterial cell. So prevents phagosome-lysosome fusion. Okay, and in here, type 3 secretion systems is going to trigger phagocytosis. Now the bacterial cell can live very happily inside the phagosome, and it can multiply. Inside the host cell, it is safe. The immune system cannot get to it. Eventually, it may break off and go into deeper tissue. Typically, that's what happens. Once it has multiplied, it will break off the host cell and begin to invade into the connective tissue, may gain access to blood, and then may become systemic. So again, type 3 secretion systems have been isolated from gram-negatives. So this seems to be a strategy that many gram-negatives have to colonize our bodies. Okay, now once the bacteria is into deeper tissue, now it's going to find the second line of defense, which is going to be phagocytic cells. phagocytic cells are going to phagocytize these are serious phagocytes that are going to phagocytize the bacterial cell. It may encounter antibodies and it will encounter complement. So once it's in deeper tissue, now we have full force immune system attacks. And the bacteria, if it can't cause an infection, it will need to fight off all of these attacks. So one strategy is just to hide within the host cell and just stay there. And it's a strategy that quite a few organisms follow. So Shigella can do that. It just stays inside host cells, jumps from one host cell to the next, and as long as it's in there, it would probably be safe. Listeria monocytogenes, which causes listeriosis, is another one that can do that. Mycobacterium tuberculosis can do that also. Then become latent and just stay inside cells. So that would be one strategy, just to hide. Another strategy would be, if he cannot hide and is susceptible to complement, then now it needs to override complement. Okay, so one of the things could be that the organism prevents complement activation. These we see in many of the gram-negatives, which are more susceptible to membrane attack complexes of complement. We call this type of bacteria serum-resistant bacteria, serum-resistant gram-negatives. They capture the complement proteins that are inactive and prevent them from becoming activated. With that, they're protected from complement attacks, from opsonization and membrane attack complexes. Neisseria gonorrhea can do that. It's oftentimes seroresistant organism. Just something here real quick. Okay. Yeah, remember we said serum-resistant bacteria are gram-negatives. And that's because it's the lipopolysaccharides of the cell wall of these organisms that oftentimes bind regulatory proteins, molecules that prevent complement activation. Okay, the next strategy would be to avoid phagocytosis. So again, one of the dangers, once the organism has crossed past the epithelial barriers and it's into the connective tissue, is that connective tissue is filled with immune system cells, like phagocytic cells. So the organism may have strategies to prevent the encounter with phagocytes. Okay, let's go back to chapter 14. And remember that in chapter 14, we said that complement C5a attracted phagocytic cells to the area of infection. Okay, so that would be one strategy, is to destroy C5a. So some microorganisms have C5a proteases. C5a peptidases. That destroys C5a. So that would be one way to deal with phagocytes, is to prevent encountering them. Another one would be to just kill the phagocytes by using toxins like streptolysins and leukocidins. Streptococcus pyogenes, which is the microorganism that causes strep throat, and it can be a normal flora of the throat. As a matter of fact, it used to be very, very common as a normal flora of the throat. It has C5a peptidases and streptolysins O that lyses blood cells. So it has both these strategies right there. Okay, so again, how can the organism prevent phagocytosis? It can prevent attachment and recognition of the phagocytic cell. Capsules oftentimes do that. Capsules prevent opsonization. Remember from chapter 14, opsonization occurs when C3b attached to the bacterial cell, and then the phagocytic cell has receptors for C3b. So the activation of C3b by the capsule itself or by specialized proteins like M proteins. Another strategy, remember from chapter 15, we said that opsonization could also happen with antibodies. So now we could have antibodies bound to the bacterial cell. And we said phagocytic cells have two kinds of receptors. They have C3b receptors and they have Fc receptors. Okay, and so the phagocytic cell binds to the Fc region of the antibody that has coated the bacteria and then eats up the whole thing. Well, what if the bacteria could bind to the Fc region of the antibody before the phagocytic cell could? So that if the bacteria has Fc receptors, it's a pretty clever strategy actually. If the bacteria has Fc receptors, now it can grab the antibody by the Fc region. And now there is no Fc region where the phagocytic cell can attach. I'll come back to this in just a second. So this is what it's supposed to happen. The antibody should bind to the bacteria by the variable region, and the Fc region is free to attach to the phagocytic cell, unless the bacteria has developed Fc receptors, which allows it to bind to the Fc region of the antibody, and now there's nothing that can be attached to the Fc receptor of the phagocytic cell. So the bacteria cannot be phagocytized. So that's a pretty clever strategy. Streptococcus pneumonia has capsules that deactivate C3b. Staphylococcus aureus has something called protein A, which is essentially an Fc receptor. Now, this last strategy, this will be the last thing we do for tonight, and then we'll wrap up the chapter next time. The last thing, another strategy, which is even more sophisticated, is Streptococcus pyogenes, which, by the way, is the same organism we said had C5a peptidases. It has a capsule with a protein called M protein that deactivates C3b. It has a protein G, which is an Fc receptor. And on top of that, some strains, not all of them, but some strains of Strep pyogenes have developed a capsule made of hyaluronic acid, which is a component of human connective tissue. So that our immune system looks at the hyaluronic acid and it leaves it alone, thinking, oh, this is just connective tissue. In reality, is a capsule that the organism is using to hide within from the immune system. So that's even a more clever strategy. It's a rare strategy. Not very many strains can do that. So match the microbial strategy with this description. Inactivation of C3b. Okay, inactivation of C3b. We said that the capsule of Strep pneumonia can do that. And we said that M protein of Streptococcus pyogenes can do that. Prevents opsonization by antibodies by Fc receptors. That was the G protein of Strep pyogenes and the A protein of Staph aureus. And then host immune system sees capsule of self molecule, hyaluronic acid capsule. That will be again Strep pyogenes. Yeah, they have a very good ability to obtain new genes. So They don't have a brain, obviously, but they have very effective ways of obtaining new genomes with genes that will allow them to do things like these, and they can pass them on from one to the next. So yes, absolutely. Yeah, and what they have for them, their advantage is numbers, because you can say it is very rare for a living thing to obtain new genes. That's true. It may happen, 1 in a billion, but when your population is 100 billion, then you can have 100 cells that are doing that. And those 100 cells are going to multiply, and within a matter of 20 minutes, you're going to have each one of them will double in numbers. So you can see how by sheer numbers, things that are events that are very rare are possible. So that's what bacteria have going for them, is numbers. So anyway, yeah, I mean, this should give you a new respect for these little guys. All right, so let's stop here. We have a little bit more to go. Actually, we're about halfway there. And we can finish it easily next time, and we'll do it after Thanksgiving. We can meet. There'll be a couple more weeks left after that. So we can meet on Tuesday again and wrap it up. We're going to meet Tuesday again, wrap this up, and then meet again on Thursday. You can meet Monday for lab, Tuesday for lecture, and then if you want to meet again on Thursday for lecture, and that way we'll just have the following week to finish up the last bit. So we should be in good shape. So we may have some extra sessions for the last week, but that's just to wrap up the last of it. Okay, any questions? Let me go ahead and stop the recording, and I'm kind of hanging there.